preparing to live stream. Can you see how many people are on the YouTube?
It hasn't started yet, so not yet.
Not yet.
Okay, now it's live.
All right. Okay. Good morning, everyone.
Just starting.
Cool. Okay. Good morning, everyone, or good afternoon, good evening, whatever you guys are. I'm Gustavo Amarante Mendes, and on behalf of the organizing committee, Karina Bortolucci, Ricardo Weinash, and myself, we are pleased to announce in a few minutes the start of the Cell Death and Immune System course, which is an initiative of 3 independent institutions here in Brazil: the Unifesp, Federal University of São Paulo, the Hospital Albert Einstein, and the University of São Paulo, the Institute of Biomedical Science. A few of you will be registered officially in one of the 3 institutions and will follow the full course, which besides this whole week of lectures by top scientists in the area, we will have also a second week of discussion, and we'll talk about that later on in the private group of emails. For now, this is a first, I have to say. Hopefully everything work well in this Zoom connection to YouTube. And I will share my screen in order to talk a little bit about the program. So hopefully, guys, please let me know if you see my screen. Are you okay? So you must have received the folder with the advertisement of the course. This is the list of the speakers who very kindly offer their time and knowledge to discuss with all of you, topics that are summarized in this next slide. So you have the time frame and all the lectures this week. Please note that Monday, Tuesday, and Wednesdays we will hold these lectures in the morning. I'm talking always Brasilia time, so you have to adjust to your time. But then Tuesdays and Fridays, the time frame change. We'll have one lecture, the 70th at the 8th at 1 to 3, and then all Friday will be in the afternoon. This is the link that you can share for the transmission of YouTube. We will be collecting questionings from all of you between the talks, and then from time to time we will stop the speakers and ask the questions. So we try to do as more dynamic as possible. Okay. On tomorrow, we have to cut very short on 12 o'clock midday, because the Department of Immunology has another lecture by Tommy Brunner in the context of our weekly seminar. So please note, and it's also will be open for everyone. Whoever is interested, just drop a note to my email. Okay. All right. So now I'd like to introduce the first speaker. Is a very good friend of mine, Seamus Martin. He's the holder of the Smurfit Chair of Medical Genetics within the Smurfit Institute of Genetics at the Trinity College, Dublin, Ireland. He has an international reputation in the field of programmed cell death, particularly, but not only apoptosis, but we're gonna hear some insights about what represents really the programmed cell death today. And remember, programmed cell death plays a key role in the development and function of the immune system, and that's part of the topic of this course. Seamus is one of the most highly cited scientists in the world on this topic. He was elected to the Royal Irish Academy in 2006, to the European Molecular Biology Organization in 2009, received the RDS Irish Times Body Medal in 2015, and this is the Ireland's highest scientific honor. Other prestigious awards include a list that I'm not going to read to you. You have it written in the screen. Seamus is a PhD graduate in the National University of Ireland and has held postdoctoral fellowships at University College London, UK, and the University of California in San Diego, where we met during the time that I was doing postdoc in the Greens Lab. Beside a long list of original and review articles, Seamus co-authored the 11th, 12th, and 13th editions of the major immunology textbooks, Essential Immunology, one of the textbooks that we use here in Brazil. Seamus is currently editor-in-chief of the FASB Journal, a leading life science journal, and the past president, actually, there was a mistake, not the actual, but the past president of the ECDO organization, the European Cell Death Organization. So thank you very much, Seamus, for joining the course. And now I turn off the screen and give it to you. Wait a second. Sharing. All right.
All right. Thanks, Gustavo. I'll just, I think you have to give me the screen because you've disabled participant screen sharing.
Okay, so let me see where it's security. Okay, now you can do it.
Okay. Okay, can you see those slides okay?
Yep.
Great. All right, good morning, everybody. Thank you for that nice introduction, Gustavo. And thanks, Karina and Ricardo, Gustavo, for organizing this course. And I'd much rather be giving this in Brazil, I have to say, but maybe in another year, hopefully, when things open up again, it would be nice to visit again. So I'm going to talk about a little bit about the background, some basic concepts in programmed cell death, why we need programmed cell death, and some of the genes involved, and a little bit of the history of the kind of development of the field. I'm going to talk about primarily apoptosis, and I'm going to contrast that with regular necrosis. I'm not going to talk about programmed necrosis. That's a field that really has, I guess, evolved in the last 5 to 10 years, and I'll maybe touch upon it at a certain point. So I work at Trinity College Dublin in Ireland, and I've worked on programmed cell death, I guess, for more than 25 years now. So as I say, I'll give a little bit of the history, and I'll start with, let me just see, I'm not, there we go. So let me just check that this is working. Okay, there's that. Seems to be just a little bit of delay. Okay, so just to start, so the body is a complex cellular society, as we all know, and the body is made up of a million cells all cooperating in more or less harmony. And just like in our own society, in a very complex cellular society, as the cells of the body basically have rules to live by. And some of the rules that cells have to live by is basically a fundamental rule is you stay where you live. Apart from certain exceptions to that, obviously cells of the immune system, which are given permission to enter various tissues in their role essentially as the body's security system. Vast majority of cells are expected to stay where they live. A fundamental rule in the cellular society, just in, I guess, our own society, is you don't eat your neighbor. Now, as we're going to see later on, in fact, that rule breaks down during programmed cell death, when cells are given permission to eat other cells in order to dispose of the dying cell corpses. So that's something we'll return to later. In the cellular society, you don't build an extension, so you don't build another property on your property without getting planning permission. So cells are not allowed to divide without permission to do so. It's very important that cell numbers in tissues are regulated. We intuitively understand that organs and all tissues have a certain fixed size. And if they go beyond that kind of limit, then essentially what we have is a malignancy, a cancer. So cells require very specific permissions in the form of growth factors and other signals in order to divide. And that's the same in human society. You can't just build an extra house on your property without getting permission from the local council or whatever. It's essential that cells do their job. If they don't do their job, then we're going to find out that there's a pretty severe punishment for not doing your job in cellular society. And that punishment is often programmed cell death. And another rule is don't allow bugs inside. So cells are not allowed to harbor viruses and other infectious agents. And then a final rule, it's not a complete list, but is don't act weird. When cells display characteristics that suggest that they're making proteins they shouldn't make, or as I mentioned earlier, starting to proliferate when they shouldn't proliferate, then again, there's a punishment for that. And the punishment is typically programmed cell death. So in a cellular society, cooperation is essential. Everyone plays their role. And as I mentioned now a couple of times, the punishment for not playing your role or for doing things that you're not supposed to do, I'm not quite sure why this isn't forwarding, is that when you don't obey the rules in cellular society, either because you're injured or you're infected or you're behaving abnormally, you're aged, or you're breaking the rules in some other way, then death is typically the penalty. And as we're going to go on to see, in most cases, the death takes place in the context of breaking the rules in cellular society is programmed cell death. So again, we intuitively understand this, that tissues are in pretty constant flux and undergo constant remodeling because of the fact that cells basically get injured, they get infected, they become transformed, and they break the rules. We know that cells die. And obviously in the adult, for every cell that dies, you need a cell to replace it. And in tissues, particularly tissues that suffer a lot of injury or infection, then there's a lot of ongoing cell death going on. And to compensate for that, there's a ongoing rate of cell division in those tissues. So there's a huge throughput of cell division and cell death to counterbalance that cell division during life. And as I mentioned a few moments ago, in our bodies, we have approximately 10 trillion cells. So 10 million cells. That's a lot of cells, all cooperating in harmony. And we turn over those cells. It's been approximated about maybe 1,000 times in our lifetime. Some tissues clearly turn over a lot faster than other tissues. So the neutrophil population is replaced in its entirety about every 36 hours or thereabouts, whereas obviously neurons in the brain live a lot longer than that. So there's a huge throughput of cells in our body, and as a consequence, there's a huge throughput of cell death. There's a lot of cell death going on in many tissues all the time. And tissues that suffer the most damage tend to exhibit the highest rates of cell death. And again, that makes really perfect intuitive sense. So if you think of the body as a box, then the surface tissues, so the surface epithelia, so the cells of our skin, of our gastrointestinal tract, the cells within our lungs, and also the cells of the immune system, that essentially get permission to go out throughout the body. These are tissues that typically exhibit a very high rate of turnover during life. And there's, as I've mentioned already now a couple of times, there's a balance between the rates of cell death and damage occurring in certain tissue and the rates of cell division. And in fact, in the adult, if we had no cell death, we wouldn't need cell division. So clearly, cell death precedes cell division in the adult. You only need to replace cells when cells have died. And in certain tissues as a consequence, so the cells that are at the most risk of damage in those surface exposed tissues, that's where we typically see the highest rates of cell death. And then in other tissues, we see very low rates of cell death indeed. And as a result of that, we see very low rates of proliferation in those tissues. So cell division and cell death are delicately balanced in the adult. And again, this is all very obvious in retrospect, but why I'm outlining this is that remarkably, it wasn't really until maybe the 1980s or 1990s that there was really an appreciation that cell death must be under some kind of molecular control. Prior to that time, cell death was really disregarded in the equation as being a force for balancing the numbers of cells within a population in mammals. So we now appreciate that cell death is regulated in many, many circumstances because of this huge amount of cell death going on in many, many tissues throughout the body. Cell death has to be regulated, really, because clearly cell death in many, many of these instances is entirely predictable. And so we see that programmed cell death is used in multiple contexts. It's used right from the beginning of life during development. Cell death is used in, for example, the sculpting of tissues. So a classic example is the formation of digits here. From an initially crude limb paddle. Digits don't just grow out from the limb paddle. What happens instead is that you overproduce cells and then you sculpt out the cells you don't want from within that crude massive tissue. And we'll see that's a theme during development. In the development of the immune system, you overproduce cells in the T and B cell compartment, and then you get rid of cells that you basically don't need or are dysfunctional. When you're making blood vessels or other hollow tubes, you make solid tubes first of all, and then you kill off the cells that you don't need. During wiring up the brain in development as well, you overproduce neurons, and then the neurons that fail to make connections are killed, and they're killed by programmed cell death. So we see in multiple situations in development, we see this overproduction of cells, and then we see programmed cell death used as a sculpting tool. So essentially, cells are told to die at a certain point of development. And that enables the body to make complex tissues. So beyond development in the adult, cell death is very important in just maintaining the fitness, as I've mentioned, of tissues, just maintaining the health of tissues. So there's a predictable rate of damage and injury in tissues. And some tissues, some cells age faster than others because of their job. And so we see cell death being simply used to keep tissues healthy. There's this homeostatic turnover of cells in lots of tissues in the adult. And particularly tissues, as I've already mentioned a few minutes ago, particularly tissues in the front line, the tissues that are most prone to damage and injury are the tissues, of course, that we typically see the most cell death in the adult. So we have cell death then used as a sculpting tool during development to keep tissues nice and healthy so that, aged and damaged and injured cells and so on don't accumulate. And then also we see cell death being used in response to damage or infection. So as I told you at the outset, there are many, many scenarios where if cells are not performing their role fully or properly, so if they're injured or they're potentially transformed, then we typically see programmed cell death is used to get rid of those cells so that they can be replaced by nice, healthy cells through cell division. So here's a little bit more detail in one of the examples I gave, developmental sculpting. You can see here in the top left an embryo. Early in development, you get these little limb bud protrusions which get progressively bigger as development proceeds. And then finally, you have a limb paddle, which becomes progressively sculpted essentially into the final form of the digits as we go around here clockwise. And during cavitation, during the production of blood vessels, as I mentioned, typically you make tubes in the body by forming solid blocks of cells initially. There's another way to make a tube, and that's by making a flat plane of tissue that then migrates inwards so that it essentially closes in itself. But a common way to make tubes is by making a solid tube of cells, and then the cells in the center become cavitated. They die through signals delivered by the cells on the outside, and those cells progressively die off in a programmed manner, again, to hollow out the tube. And that's seen in many situations during development. For those of you who are studying immunology, I know a lot of you looking at this today are immunologists, and you'll know that during selection of T cells, essentially what you do is, because you're trying to make receptors, T cell receptors against antigens that aren't present in the body at that stage. So you randomly recombine T cell receptors, and then you essentially delete all the T cells that make receptors that either completely fail to recognize MHC, so they don't work at all, or receptors that too avidly recognize MHC, so they're autoreactive. And this form of negative selection or death by neglect is typically programmed. And it's really happens at an astonishing rate in the thymus during development. It's been estimated that over 95% of developing T cells are killed through programmed cell death. So it's an extremely wasteful process in many ways, but when you think about it, there's no other way to really fine-tune the T cell repertoire against antigens that you haven't yet experienced or haven't yet seen than by making all possible combinations and getting rid of all combinations that are potentially autoreactive. So, programmed cell death enables the sculpting of the T and B cell compartments in a similar manner in order to get rid of all the cells that you don't need. And another example that we'll look at a little bit more in detail in a little while, cytotoxic T cell killing or natural killer cell killing for cells that are either infected with viruses or bacteria or potentially transformed. Again, it's very, very useful to be able to simply kill that cell as a means of shutting down the infection and ensuring that the infectious agents cannot override the programmed cell death mechanism within the target cell. So typically we have professional assassins essentially in the body that if the target cell itself can initiate death of itself, then the cytotoxic T cell or natural killer cells will deliver a kiss of death, either in the form of what are called death receptor molecules, death ligands that engage with death receptor molecules on the target cell, or through delivering cytotoxic granules into the target cell. And we'll look at the detailed molecular mechanism of these pathways in a little while. So I've given you a few examples of situations in which it's very useful to be able to kill cells in a regulated manner for, as I've said now, for reasons of simply sculpting tissues during development, for keeping tissues nice and fit and healthy in the adult, and also in disease situations to get rid of cells that are potentially going to either cause a cancer or infect the body with a virus if those cells are allowed to stay alive. So we know in all of those situations, being able to kill cells on cue is of course extremely advantageous. So it's obvious that there must be some kind of molecular mechanism that regulates this. And of course, when this mechanism fails, disease almost always results. I've already touched upon cancer. So we know now that cancer develops, or cancer can arise as a result of a failure to kill cells that are on the way to transformation. And Christina will talk about that later in her talk. But of course, in the immune system, we know that if you fail to get rid of potentially autoreactive T cells or B cells, then autoimmunity frequently develops. And this is an example of, for example, a wild type mouse. Well, in fact, this is a heterozygous mouse that has one copy of the FAS receptor, and then where there's a full knockout of the FAS receptor. And the FAS receptor is very important in homeostatic turnover of T and B cells in the peripheral compartment. So what typically you get is you get an accumulation of these extra T and B cells, and you can see here that the normal lymph nodes of mice here on the top are enlarged about 50-fold within about 4 months of birth in a fast knockout mouse. So FAST is involved in culling peripheral T cells and B cells, and if you can't regulate homeostatically the T and B cell compartment, then you get a massive expansion of those cells and an accumulation of those cells. And as I say, the result is many more extra T cells and B cells, and you see massive enlargement of the lymph nodes and spleens. And these mice then typically develop a disease not unlike systemic lupus erythematosus. And there's lots and lots of situations where if cell death becomes dysregulated, then the consequences are basically pretty severe. As I've said, autoinflammatory disease or autoimmune disease is a common outcome, or cancer in many cases. And on the other side of the coin, if you have accelerated cell death, so cells dying in response to stimuli that normally wouldn't kill them, then typically we see an accelerated loss of cells. And I think this is particularly important in long-lived cell compartments such as neurons, where neurodegenerative diseases seem to rise as a result of dysregulation of the normal rates of programmed cell death that take place in the brain. So let's move on. First of all, I'm going to talk about phenotypically what apostatic cells look like and necrotic cells look like, and the basic events that take place in those cell death modalities. And then I'll talk about what I think is essentially the rationale for having programmed cell death, because of course I've mentioned lots of situations where it's useful for cells to die, but of course cells could just die by necrosis in those scenarios. So we could have some kind of way of just shutting off a cell, and then when we need to kill a cell, the cell just bursts open by necrosis, and that indeed does happen in certain special circumstances. But in the main, when we see regulated cell death occurring in all of the contexts which I've just talked about, typically it takes the form of a type of programmed cell death called apoptosis. In fact, this are some images we took in my lab a couple of years ago. And these cells here that are in red and have these very intensely stained nuclei, this is Dappy Stain. You can see the nuclei are broken up into very intensely stained small pieces. And this is very characteristic of an apostatic cell by comparison with a healthy, viable cell. You can see here on the bottom or on the left-hand side where the nucleus is very large. It encompasses almost the entirety of the cell body. Apostatic cells, by comparison, condensed or blebbed, and you can see very intensely staining nuclear fragments in the cells. And we get to discuss why that happens and how it happens. So I'm going to go through essentially the mechanism of programmed cell death, and consider what's the kind of main functional driver behind this mechanism. What's really going on here, apart from, of course, killing the cell, but what are the major considerations we need to take into account when when cells are dying? So if I may play this for a moment, years ago, we used to think that, when cells died, they went around their own business, and then suddenly one day something, really horrible happened. And then that was the end of it, essentially. And that's what I call the car crash view of cell death, where until really the 1980s, if you read about cell death, you read almost invariably, about necrosis. Cell death was necrosis. Pathologists who study cell death or look at cell death in tissues invariably called cell death necrosis. There was one form of cell death. Nobody considered that it was kind of regulated or programmed in any way. And it was considered that cells effectively, at some point ran out of road or hit a wall and just fell apart. And of course, necrosis does take place. And I'll tell you that there's situations in which it does take place, but we know that in the main, the vast majority of cell death that takes place under the situations I just spoke about a few moments ago, during development and homeostatic turnover of tissue, and even in the disease scenarios such as viral infection or on the road to cancer, we know that most of those cell deaths don't take the form of necrosis. And I'm going to tell you some of the reasons why I think they don't take the form of necrosis. So I'm going to be talking about necrosis, which you could consider as kind of cell murder, in the sense that the cell, when it undergoes necrosis, doesn't really choose to die or participate in its own death in the main. Now, again, I must add a caveat here that there are certain types of programmed necrosis that have been discovered in recent years. These types of programmed necrosis typically happen in immune scenarios where cells, where it's advantageous for cells to release its contents very explosively. And I would say that they're important exceptions to what I'm going to tell you today. And in fact, I think underscore a major point I'm going to tell you today, which is one of the main driving forces behind having a programmed cell death is to avoid inflammation. To avoid inflammation and to avoid collateral damage of cells around. But in certain exceptional cases where you absolutely want inflammation, then you typically see programmed necrosis. But that's a special exception. In most tissues, when a cell doesn't choose to participate in its own death, I'm talking here about situations such as, for example, you suffer a blunt trauma injury. You whack your hand against something, or you're hit by something, or you get a burn. When cells are overwhelmed by a nasty stress or exposure to damage, then that's necrosis. And typically cells simply burst open, release their contents. It all happens very quickly. I want to show you some examples of this. But when cell death is regulated, this is more of a cellular suicide, where events are under the control of the cell that's dying. And as we'll see, there's actually a molecular machinery that's controlling the death of the cell. In most of these cases, the cell senses that it should die. It switches on its own programmed cell death machinery. And in those cases, cells die by apoptosis, which is this form of regulated cell death I'm talking about. And then there are special cases, I've mentioned them already, where the cell doesn't necessarily switch on its own machinery, but another cell does that job for it, possibly because there's something wrong with the cell that has paralyzed its own cell death machinery. And that's cytotoxic T cell killing, or natural killer cell killing, where the cell is infected by a virus. The viruses frequently encode inhibitors of programmed cell death, of course, because viruses want to keep the cell alive for as long as possible. And or the cell may be transformed, and in the process of becoming transformed, it suffered damage or an activation of genes that regulate cell death. And so in those cases, you have a form of assisted suicide, where a cytotoxic T cell or natural killer cell will execute a cell by turning on its programmed cell death machinery, as we'll see, by delivering things into the cell that's capable of doing that. So that's another special form of apoptosis that very often people overlook when discussing programmed cell death. So I'm telling you that I'm going to talk about events that are similar but different. This is an old joke, actually, but I do think it kind of vaguely represents the point I'm trying to make here. So this person is not dying, in fact, is meditating, and it's an advanced form of meditation where you go in this position for several hours at a time. And this person is in a very similar position, but there's no meditation involved. And the kind of upstream stimulus that got him into this state are very different from the upstream stimulus that got this person into a vaguely similar state. So what I'm going to say is that in some respects, yeah, dead is dead. If a cell dies by necrosis or apoptosis, it ends up dead. And superficially, that's the same endpoint. But actually, the upstream drivers and the kind of means to get to those endpoints are very different in the two forms of cell death that I'm going to tell you about. So I've alluded to this point earlier. So I've told you lots of situations where it's important to be able to kill cells in order to keep tissues healthy, in order to sculpt cell populations, to get rid of cells you don't need during development. And also then in disease situations where the cell's infected or potentially transformed. The point is, you might think, well, it doesn't matter how the cells die. As long as you get rid of the cell you don't want, that's the important thing, right? Well, no, that's not quite right. It's far better, if you want to get rid of a cell, that you don't allow that cell to burst open and spill out its contents. And so one major impetus for having a regulated form of cell death is to control the disposal of cells so that they don't simply burst open and release their contents. And there's two main reasons for this. The first reason is cells have lots of nasty enzymes inside them. So I've noted some of them there. Of course, cells have lipases that hydrolyze lipids, and they would be disastrous if they were released into the extracellular space. They'd start hydrolyzing lipids in neighboring cells and bursting those cells as well. Cells have proteases, carbohydrases, DNases, RNases. DNA itself is cytotoxic, and it's a very viscous polymeric molecule that would cause a lot of problems. Not to mention lots and lots of other molecules that are normally compartmentalized in cells. So you don't want that being all released into a tissue because it's going to cause collateral cell death and, essentially cause a big mess and be very wasteful. So if you're going to kill cells in all the situations we've mentioned, it's of course better that they don't just simply burst open and die. Now in necrosis, cells, that's pretty much what they do. They burst open, they release all their cellular contents. And typically when you see necrosis in a tissue, it doesn't affect just single cells. You typically see hundreds or thousands of cells that have undergone necrosis because of this issue of collateral damage. So does it matter how cells die? Yes, it does matter. Dead isn't dead. There's different kind of degrees of death in the sense that it's far better that if a cell's going to die, it somehow signals that fact to other cells so they can kind of prepare to deal with the disposal rather than be simply subjected to a cell rupturing and bursting open and just releasing the contents and causing, obviously a lot of discomfort and damage to the neighbors. And another reason why it's really important to regulate death of cells is that the immune system responds very differently to the presence of necrotic cells, which are the ones, as I mentioned, that simply burst open and rupture and release their contents, and cells that have undergone programmed cell death, cells that have undergone apoptosis. The immune system essentially ignores apostatic cells, immunologically speaking, and makes a very strong response to necrotic cells. And this notion that necrosis is somehow seen in a special way by the immune system, this wasn't appreciated until probably the mid-90s when an immunologist called Polly Matzinger wrote a very important review article at the time, I think it was in 1994, where she introduced the concept that the immune system doesn't really recognize non-self as we often think about as immunologists. So Janeway and others suggested that the immune system is tuned towards non-self, and to some degree that's true when you consider the kind of molecules that activate PAM receptors, which are innate immune receptors. But it's not completely true because we know that, for example, if you take a non-self peptide, so a completely foreign peptide that our immune system has never seen, and inject that into the body, typically you don't get an immune response. The immune system completely ignores it. And it ignores it unless it's usually accompanied by evidence of danger. And Matzinger kind of considered this and really dwelt upon this and introduced the notion that the immune system responds not to non-self per se, but non-self in the context of danger. And so danger can either be detection of a PAMP, a bona fide pathogen component, which is clearly something that has been tuned to bind to PAMP receptors. So the immune system certainly recognizes that as dangerous. But if you see a peptide in the absence of a PAMP, there's another scenario which may generate an immune response, and that's if the peptide is seen in the context of necrosis, which she put forward this idea that necrosis was recognized as danger just as much as a PAMP is recognized as danger. So the immune system responds to detection of a bona fide pathogen component or detection of necrotic cells. And in either of those scenarios, then if you present non-self peptides, you will get an immune response. So Matzinger realized that there must be molecules released from necrotic cells that are just as potent as PAMPs in engaging the immune system. And essentially that's, called the danger model of immunity. Now, she didn't know what those molecules were. This was, a proposition that she put together. And it turned out that she was correct, that necrotic cells are capable of activating the immune system just as much as PAMPs are. The molecules that she posited that are released from necrotic cells, she called danger signals. They're now called DAMPs, or damage-associated molecular patterns. So there are two forms of patterns, if you like, that are very potent, that engage the immune system: pathogen-associated molecular patterns, or PAMPs, and damage-associated molecular patterns. These are molecules that are released from necrotic cells. And so I'm going to tell you in a couple of moments what I would consider to be classical DAMPs. But the point here is it doesn't matter whether a cell dies by apoptosis versus necrosis, because what we see is that necrotic cells typically release DAMPs, whereas apoptotic cells don't release DAMPs. In fact, I would go so far as to say that the major raison d'etre for apoptosis is essentially to prevent the release of DAMPs in a tissue, to make sure that when cells die, they don't activate the immune system by releasing DAMPs. So many of the things that happen during apoptosis are about ensuring that cells are essentially recognized early enough that they can be removed and engulfed by phagocytes or neighboring cells, so they don't release DAMPs and they don't drive inflammation and they don't provoke collateral cell damage within a tissue. So that's just a concept there. Necrotic cells, they release DAMPs that activate cells, the immune system, and that's something that we really want to avoid during programmed cell death. So, of course, healthy cells, they're ignored by the immune system. Necrotic cells, they're dangerous. So you want the immune system to make an immune response in the context of necrosis, because, as I mentioned earlier, typically you see necrotic cell injury really in very severe cases where you've had, experienced really quite severe trauma. And the kinds of things that induce necrosis are the things really the immune system wants to know about. If you have a burn or a major compression injury in a tissue that's killed thousands of cells, that's provoked necrosis. That's a scenario where you want the immune response involved or the immune system involved, because, that's a serious injury, which is almost certainly going to be rapidly accompanied by infection. And so you want the immune system to go in there. And also, you want the wound healing properties of the immune system in order to kind of heal that kind of damage. But if cells are dying by apoptosis, if this is just during development where you're getting rid of some cells because, you want to sculpt out a tissue, that's not a scenario where you've got injury or infection necessarily, or any major damage that the immune system, should know about. So typically in apoptosis, I think many of the events are conspiring towards minimizing, the activation of the immune system, so that the tissues are not in a constant state of immune activation. Because, as I said at the very beginning, of course, apoptosis takes place in this homeostatic way at a huge scale in many tissues throughout life. So this is a theme I'm going to return to a number of times during this talk. Effectively, that apoptosis is immunologically silent as a result of the production of what have been called find me and eat me signals. So during apoptosis, what we see happening is cells display things that make them look different to the immune system, and they even secrete certain molecules, certain signals that facilitate early recognition of apoptotic cells by the immune system. And so typically what we see is apoptotic cells are eaten by their neighbors, or they're eaten by phagocytic cells. And as I said at the very first slide, that's a fundamental rule in a multicellular organism. You don't eat your neighbor, but that breaks down during apoptosis. Apoptotic cells give permission to their neighbors and also to other phagocytes to basically eat them. And that achieves two goals. It prevents them from leaking open, releasing DAMPs. And of course, it prevents the cell from killing its neighbors as a result of release of cytotoxic enzymes. And of course, it's a much easier way to recycle cellular constituents by allowing another cell to engulf those, digest them, and recycle the contents, of course. So typically what we see is apoptotic cells are either non-inflammatory or actively anti-inflammatory. There seem to be certain signals released by apoptotic cells that, in fact, make macrophages and cells that engulf them that convert them into an anti-inflammatory state. Whereas when cells die by necrosis, They, as I've said now a few times, they release DAMPs, as suggested by Matzinger, and those DAMPs bind to DAMP receptors and actually, in fact, promote inflammation. So what are DAMPs? Well, that's something that's been much debated in the kind of 20 or so years since the concept of DAMPs was introduced. And I would say that's still a picture in evolution. You know, we still don't know the full spectrum of DAMPs. But I think most would agree that members of the IL-1 family represents, let's say, canonical DAMPs. So these are cytokines that we've worked on, and in fact, we propose that members of the IL-1 family really are the key canonical DAMPs that are the classical example of a cytokine that is not normally released from cells, that is nice and healthy and alive, and in fact is only released during necrosis. And it's something we've worked on for the last 10 years or thereabouts. There are other molecules that have been proposed to be DAMPs, HMGB1, DNA, ATP. I will say that some of these molecules are controversial, but it is known that, you know, that in certain scenarios, all of those molecules, when released from necrotic cells, have the capacity to activate the immune system in one form or another. So the concept is, as I've said a few times now, when cells die in response to overwhelming injury, they undergo necrosis, they release these DAMPs, and there's members of the extended IL-1 family here. And then those DAMPs go on to ramp up the inflammatory response, bring in cells of the immune system, obviously, into tissue, because when you have necrosis, you've got very significant tissue injury. We think IL-1 family members are really perfect examples of DAMPs because of several reasons. They're all bona fide cytokines, so there's absolutely no dispute about whether these are cytokine molecules or not. These are cytokines, but they lack secretory signals. So the vast majority of interleukins, for example, they all go to the Golgi, the Golgi secretion pathway, and you know, cells, they have specific secretion signals, and it's very clear how they get out of the cell. But IL-1 family members are an exception. They have no secretory signals. And in fact, it's been debated very much for many years as to how they get out of the cell. And it seems to be that the primary route out of the cell is when the cell dies by necrosis. And there's certainly no doubt that they all act at the apex of cytokine cascade. So again, this is very desirable and DAMP if you want to drive or ramp up inflammation as a result of necrosis. And it's very good that the cytokine you release is able to initiate lots of other aspects of inflammation. And virtually all IL-1 family members are capable of triggering the secretion of numerous other cytokines and chemokines, and essentially initiating all aspects of inflammation. And of course, again, another qualification of these cytokines is they're all enriched at barrier tissues, gastrointestinal tract, the skin, the tissues that are most prone to injury and where you'd really expect to see DAMPs enriched. So I'm going to skip through some of the discussion on DAMPs. I was just going to give you further evidence as to why we think DAMPs or IL-1 family members serve as the canonical DAMPs. But, you know, I'll just leave it that, you know, we certainly think the members of the extended IL-1 family represent really important DAMPs, and they get activated by a variety of mechanisms. But we think one of the major mechanisms that get activated is that by the fact that when you get necrotic injury, typically within minutes of getting necrosis, you get literally millions of neutrophils migrating in here to the wound. So this is an example of a sterile injury. It's from a paper by Tim Lammerman and colleagues. And what they did was they used a laser to sterile injure, to kill a group of cells. And so here it's a complete sterile injury. There's no infectious agents present. And then the neutrophils here are labeled with FITC, and you can see here they swarm in. And within less than an hour, you have an absolute swarm of neutrophils, you know, surrounding the necrotic tissue. And we know that when neutrophils are confronted by necrotic cells, typically they release a whole host of proteases they have in their specialized granules. We've worked on this for a number of years and shown in a series of studies that the neutrophil proteases that are released at the site of tissue injury which include elastase, cathepsin G, and proteinase 3, they're all very good enzymes at processing multiple members of the extended IL-1 family. So we think these things go very much together. When you get necrosis, you very quickly get neutrophils on the site. Neutrophils bring in proteases, and those proteases process and activate multiple members of the extended IL-1 family. And of course, famously, Caspases also process two members of this family, IL-1 beta and IL-18. And I think, you know, caspase-mediated processing of IL-1 beta and IL-18 is very important in the context of regulated necrosis or programmed necrosis, but I'm not going to dwell on that in this talk. You'll hear about that later in the week. So going back to the main point, necrosis or necroptosis, regulated necrosis, is highly inflammatory because of the release of DAMPs from cells. And typically when you get necrosis, you get rapid cell rupture, release of DAMPs, and then those DAMPs ramp up immunity. Whereas by contrast, apoptosis is non-inflammatory because apostatic cells, they display signals that enable neighboring cells to recognize them early. So you don't get DAMP release, cells don't rupture, and they don't drive inflammation. And that's the kind of key functional difference between having programmed cell death and a non-programmed cell death, in my opinion. So I've mentioned this now, and again, you'll have a talk on this later in the week, that phagocytes are capable of recognizing apostatic cells and engulfing them and removing them from a tissue before they release, before they burst open and release DAMPs. And I'll mention a little bit about the mechanism of that later on. So let me just go through some features, some morphological features of unregulated or non-programmed cell death, and then contrast this with programmed cell death. So I mentioned it several times now. So necrosis is akin to cell murder, where the cell doesn't really participate in its own death. It doesn't choose to die. It's typically seen in response to really severe trauma, rupture, compression injuries, toxins, large amounts of alcohol, pore-forming proteins that certain infectious agents bring to the cell, or extremes of heat or cold. High-dose radiation is typically something that can cause rapid necrosis. So typically things that kill cells very quickly, within minutes, or certainly less than an hour, really nasty stuff, is typically what provokes necrosis. And you know, in necrosis, what we typically see is, you know, and these sequence events happen usually within, 30 to 60 minutes. I'm going to show you a movie of this in a moment. Cells typically round up very quickly. They undergo high amplitude swelling. All the organelles swell in the cell, including the nuclear compartment, and then the cell ruptures and releases its contents. So this is really a very unfriendly mode of cell death, as I've mentioned now multiple times, and it releases danger signals and activates the immune system. So this is a movie we made in the lab a number of years ago, where these are cells, HeLa cells, and this movie is made over a period of an hour. And you can see here that the cells have been exposed to high dose hydrogen peroxide. What you're seeing is high amplitude blebs appearing very rapidly in the cells, and the cells are just bursting open, releasing their cell contents into the extracellular space. And all cells in the dish, are dying really in synchrony within minutes of each other. And that's typically what you see when cells die by necrosis, that kind of pretty much everything dies all at the same time. I'm just going to show that again. So again, here's the beginning. The nuclei look fine. There's the first cell that's ruptured there. Then the nucleus is more prominent. There's another one. You see these blebs are going up very, very quickly within a short space of time of one another. And not much else happens really. And necrosis is kind of a relatively uninteresting, from the point of view of the cell behavior that's observed. And this is very different to what we see during apoptosis, as I'm going to now show you in a moment. So this is kind of necrosis. This is a movie that we made at a meeting a few years ago. Things that cause necrosis are just very sudden events, very overwhelming events, and many cells die altogether all at once. There's really no time to react. And in fact, that signal would be very much a type of necrotic trigger. So in apoptosis, as I've referred to it a few times now as a type of cell suicide, it's very, very different. The cell makes a decision to die itself. Typically, the triggers of apoptosis are natural triggers made by the body itself. They may be, in fact, just cell signaling molecules like TNF or fast ligand or trail ligand, or just cytokine deprivation. Removing a molecule like IL-2 from T cells will trigger programmed cell death. So not necessarily injurious signals in any way, but also injurious signals that provoke mild damage or injury also can provoke programmed cell death. And typically what we see in programmed cell death scenarios is that the cell takes many more hours to die than it does by necrosis. So we often see cells undergoing apoptosis 12 hours after they've been exposed to the stimulus, or even 24 hours after, or sometimes even longer. But if a cell has to die, this is definitely the way to do it, because it's a much friendlier form of cell death, and it limits activation of the immune system, as I've already mentioned now a few times. So where does this term come from? Well, of course, scientists had recognized as cells with apostatic features, which I'm going to discuss in detail in a moment, cells that had these condensed nuclei and fragmented nuclei. Scientists recognize these from the late 1800s. There have been kind of scattered instances in the literature of people mentioning cells that had characteristics of what we now know are apostatic cells. And they were called various things, sunburn cells and other names. They were really considered oddities, or maybe, things that happened under very special circumstances. But there was a paper published in the Annual Review of Cytology in 1972 by a group of pathologists working in Edinburgh, led by Alistair Curry. And the first author of that paper was John Kerr. The middle author was Andrew Wiley. And Andrew Wiley kind of took up the torch in this area a bit later on, because actually this paper was completely ignored by the scientific community for, I guess, maybe 20 years. And what this paper did was introduce the term apoptosis for cells that were dying in lots of tissues that they had studied in this paper. It's largely a paper focusing very much on morphology. And Kerr, Wiley, and Curry realized that when cells died in many different tissues, there seemed to be a kind of a common phenotype. Cells all looked very similar. And they also saw that in many instances where cells died, what they suspected was a kind of a regulated or programmed manner. They suspected that there was a common machinery at play. So there was, this was a regulated form of cell death. Now, they had no evidence for this in this paper. They hadn't a shred of evidence. The only evidence they had was that those cells all kind of looked broadly similar. And also they noticed that very often those cells with that phenotype, which they call the apoptotic phenotype, for reasons I'll explain in a moment, they saw those cells within phagocytes. They often saw those cells within neighboring cells. And in fact, that paper published by Kerr et al. In 1970 is remarkable for its prescience, in that they laid out the whole process, but they had very little evidence for this. But they suggested that when cells were dying with this morphology, and the morphology they identified was cells with highly condensed DNA and fragmented DNA, and cells that had bleps and so on. And they proposed that these cells represented cells undergo programmed cell death. They wanted to introduce a kind of a fancy scientific term for this. So they went to a colleague of theirs, a professor of Greek, I think in the University of Dundee, who suggested the term apoptosis, and it kind of derived from the Greek for ptosis, meaning to drop or fall away. And they had actually, they had kind of described this process to their colleague as akin to how, the leaves fell from trees in autumn. So it wasn't signifying that the tree was dying, but merely that it was replenishing parts of itself. And they suggested that when cells were dying in this manner, it meant that the tissue was just replenishing itself. It was akin to this process of petals falling from flowers or leaves falling from trees. And so that's where the word apoptosis comes from. In that paper by Kerr and colleagues in 1972, they introduced the term apoptosis to represent cells that were undergoing programmed cell death with the characteristic features of apoptosis, which I'll show you in a moment. And but it's a remarkable paper, I think, for its kind of vision, in that based on very little molecular evidence, they proposed that these cells represented cells that were undergoing death in a programmed manner. And certainly, if you're interested, I would certainly strongly recommend you reading that paper. So these are the characteristics that they identified in the paper. This is not from their paper, it's from a paper from my own lab. But essentially, what you have is, if you look at healthy cells, of course, and you stain the nucleus here in the top middle, the nucleus is stained with hooks. And you see nice decondensed nuclei, and they just look like you'd expect a nucleus to look. Whereas when cells start to undergo apoptosis, the nuclei look really very, very strange. And this is the kind of characteristic that Kara and colleagues were really struck by, that these cells that seem to be undergoing cell death in this programmed manner, they had highly condensed nuclei. The nucleus was often broken up into several pieces, which again is very strange. And also the apoptotic cells always had these kind of multiple blebs on their surfaces that were not unlike kind of a bunch of grapes-like appearance, and with very often the piece of nuclei within these blebs and so on, as you can see on the right-hand side. By contrast, necrotic cells, the nuclei in necrotic cells don't look very different really to healthy cells. They swell a bit, there's maybe a little bit of condensation, but you don't have these highly condensed and fragmented pieces of nuclei that you see in apoptotic cells. There's just some examples of some of the differences. So I've mentioned about the blebbing. Left-hand side here, you have a viable cell. On the right-hand side, this is an apoptotic cell with lots and lots of blebs being produced. These are now called apoptotic bodies. And another thing that was found a few years later by a paper published in Nature by Andrew Wiley in 1980 was that, strikingly, apoptotic cells exhibited this ladder-like pattern of DNA hydrolysis when the DNA was analyzed by agarose electrophoresis. So if you take a healthy cell or healthy cell population and extract the DNA, run it on an agarose gel, you find it's all high molecular weight. But with apoptotic cells, you get this progressive fragmentation. So this is kind of the first molecular marker of apoptosis. And we now know this as a result of activation of an endonuclease, which hydrolyzes DNA in a promiscuous way once it's activated. And when that happens, you just get hydrolysis of DNA to internucleosomal sites. And because there's about 200 base pairs wrapped around the nucleosome, this protects the DNA from being hydrolyzed. And so you get this 200 base pair or internucleosomal-like ladder pattern of DNA hydrolysis during apoptosis. And that, of course, contributes to the fragmentation of the nuclear DNA and the condensation of the nuclear DNA, this hyperpyknotic collapse of DNA that's seen during apoptosis. So here's the events in apoptosis. Stepwise, you get a cell, if it's attached to a substratum or neighbors, it will typically detach from those neighbors, round up. Then it will condense. It usually, in the earlier phases, actually gets smaller. So this is the opposite to necrosis. Cells normally expand and enlarge and swell during necrosis. In apoptosis, we typically see kind of a shrinkage and a blebbing, and the nucleus, becomes highly condensed and it starts to fragment. And then we see the production, the shedding of these blebs into apoptotic bodies. And of course, in vivo, well before even you've got the production of apoptotic bodies, macrophages recognize cells, even at this very early stage, they recognize cells as being apoptotic due to changes that take place on the surface of the apoptotic cell early on. And those phagocytes or neighboring cells engulf the apoptotic cell and remove it well before it has a chance to burst open. So typically, apoptotic cells don't burst because they display changes that enable them to be recognized by their neighbors. To break that rule that you normally don't eat your neighbor, well, in the context of apoptosis, you do. So dramatic reorganization of the cellular architecture occurs during apoptosis. And all of this, we think, is kind of facilitating the controlled dismantling of the cell so that it can be disposed of in an efficient manner and facilitate its removal with the minimum of disturbance to neighbors and without releasing dams and activating the immune system. So this is what apoptosis looks like, again, in the lab. This is a movie that we made in the lab a number of years ago. And these cells are dying over 24 hours. So the movie I showed you of necrosis was cells dying, the same cells dying in response to a necrotic stimulus that all died within an hour. Whereas, we're probably about 8 or 10 hours into the movie now. And you can see that the same stimulus that caused this cell to start dying first, this cell is completely withstanding for many, many, many hours longer. And so is this cell. And these cells are, in the various stages of apoptosis. You can see it's much more active than necrosis. The cells have rounded up, they're undergoing blebbing. Now the blebbing is becoming high amplitude, and that's secondary necrosis. But of course, in vivo, typically cells wouldn't undergo secondary necrosis. They would be removed at the early stage. For example, that this cell is at, we already know that it's displaying the membrane changes that are typical of an apostatic cell that enable a macrophage to recognize it and engulf it. So I hope you'll agree, by comparison, the changes that take place in apoptosis are much more dynamic than that take place in necrosis. And it's really a rather beautiful process when you look at it. Cells are initiating all of these changes. We know that the molecules that coordinate most of these events are caspases. They're a family of proteases, which I'm going to get to. And caspases really, now they're active at the earliest stage of apoptosis, and they're cutting up molecules and they're instigating all of these changes. And we know that because if you block caspase activity in response to an apoptotic stimulus, then you effectively convert the phenotype to necrosis. So we know that caspases are coordinating events in the cell that produce all of these apoptotic changes. And again, it's a point I've made a number of times now, but it's a really critical point. Really the major functional driving force of apoptosis is to facilitate recognition and removal of the dying cell before it is an opportunity to burst open and cause damage to surrounding cells and release stems. And the reason why apostatic cells are recognized, you're going to have, I think, a full lecture on phagocytosis of apostatic cells and the consequences. There seems to be a number of so-called eat me signals that are displayed. Phosphatidylserine is probably one of the most important ones. Normally, phosphatidylserine is it's confined to the inner leaflet of the plasma membrane, and during apoptosis, it's externalized. And also, there are molecules that have been implicated as find me signals that are secreted by apostatic cells, and that enables phagocytes to find them and home in on apoptotic cells early before they burst open. And phosphatidylserine can be conveniently stained using an XM5. And I mention that because it's an assay that's widely used for the detection of early apoptotic cells. And we know that cells externalize phosphatidylserine very early in the process of apoptosis, well before you've got significant blebbing, and certainly before the cell has started to leak open. And using FITSI labeled an XM5, you can detect phosphatidylserine on the surface of apoptotic cells. I'm just illustrating this here to show you a little bit of data. If you stain cells with an XM5, normally cells don't bind an XM5, and this is propydium iodide, a dye that detects essentially cells that have leaky membranes, essentially necrotic cells. So in a healthy population, cells will be PI negative and an XM5 negative. And then if you trigger cells to undergo apoptosis, these cells have been stimulated with actinomycin D. Here, after 6 hours, you can see almost 50% of the cell population are now binding an XM5. So they've externalized phosphatidylserine, but they're not taking up propydium iodide, so they're not leaky. This change is happening very early before cells are entering anywhere near secondary necrosis. And then even later, 12 hours, virtually all the population now is externalized phosphatidylserine, and only a small minority of the population now have started to leak. There are cells that probably even before the experiment started were already on the way to necrosis. There's always a background level of cell death happening in culture. But the point is, cells externalize phosphatidylserine, which is an eat me signal, which gives permission for phagocytes or neighboring cells to eat the cell, which again, breaks a fundamental rule of multicellularity that you don't eat your neighbor. Well, you do during programmed cell death. So again, it's a point I'm going to say a couple more times in this lecture. Apostatic cells are immunologically silent, whereas necrotic cells are not, again, due to the display of signals that facilitate their uptake by phagocytes prior to the release of DAMs, which is what really I think is the kind of functionally defining characteristic between apoptosis and necrosis, the release of DAMs or not in those two modes of cell death. Now, I mentioned earlier the cell execution. So we have cell murder, which is necrosis, cell suicide, which is apoptosis, and then cell execution is involuntary apoptosis, where a cell hasn't been able to turn on its own cell death machinery. And, I mentioned earlier that this is, typically in the context of natural killer cell or CTL killing, where you have these specialized immune cells that turn on the programmed cell death machinery. And again, we know exactly in molecular terms how this works now. And there's specific sets of receptors as well as cytotoxic granules that are delivered to the target cell. I'll mention this later. Just want to show you an example of this. These are, again, HeLa cells, and these are NK cells that have been added to the HeLa cells. And the NK cells are the ones moving around. And what you're going to see is this NK cell here is going to attack this cell, and it's now degranulating. And this cell is going to undergo apoptosis. And the neighbors will be completely spared. And so this cell should now start to initiate apoptosis. So there we go. So that's a nice example of this type of assisted apoptosis, or you see this cell, this NK cell is kind of going crazy. I presume it's detected the degranulation and it's crawling around the culture in an agitated state. So that's an example of NK-mediated killing, where you've got this kind of assisted suicide, and typically we, I think, see that in the context of either cancer or a viral infection. So I've told you about why do you need programmed cell death? I've told you about well, what's desirable in a programmed cell death mechanism. I've told you it's desirable to avoid collateral damage by releasing cell contents, and that it's desirable to avoid provoking inflammation by releasing cell contents, i.e. DAMs. But how does it all work? So let me discuss some of the molecules. I've mentioned now caspases as being proteases that are activated during cell death. So there's a very sophisticated molecular mechanism that's been identified through work by lots and lots of labs over the last 20 to 25 years. So I mentioned that, prior really to the late 80s, even though the paper on apoptosis was first published in 1972, that was largely overlooked by the community. And then in the late 80s or early 90s, the scientists, and I think largely immunologists, began to realize, cell death must be programmed in lots and lots of situations. And if it's programmed, then there has to be a molecular machinery. And so they start looking for the genes involved. Now, I'm going to actually just go through the genetic control, molecular control of cell death. And I'm going to start off in the primitive nematode, Cinerabdos elegans. And then I'm going to consider the same machinery in the fly, Drosophila melanogaster. And then I'm going to move on to compare that with mammalian cell death control. But I don't want you to think that all the genes were discovered in the nematode, Cinerabdos, and then the same genes were discovered in the fly. And then, that was all then transferred to mammals. It wasn't like that. So the understanding of molecular control of cell death, was worked out by, different groups of people working in mammalian systems, so on human cells and mouse cells, and then people working in nematode systems and people also working in the fly. And they all kind of made their contributions. And in fact, this was in the early days, in the 90s and later, it was all kind of very messy for a while because, it wasn't clear exactly how this all fit together. And in fact, as I'm going to describe to you, there are some notable differences, very noticeable differences between how programmed cell death works in the nematode versus how it works in the fly and how it works in mammals. But as you're going to see, there's also a lot of similarity as well between the three systems. So let's look at this. So the nematode Cinerabdos elegans is a very useful model organism for doing genetics. It's a relatively complex organism. It's about 1,000 cells, lots of specialized tissues. And The use of the worm was pioneered by Sidney Brenner, working at the Laboratory of Molecular Biology in Cambridge in the 1970s. And he reckoned that it would be useful to have a complex multicellular organism, that wasn't too complex, such as a fly, for example, or a mouse, to do genetics. Because up to that time, people had been largely doing genetics on bacteria and yeast. And so he championed the use of the nematode. In fact, it was originally Cinerabdos briggsei he started working on, but for various reasons, Cinerabdos elegans was the nematode that ultimately was chosen to be the model multicellular organism for doing genetics. And at that time, Brenner had in his lab two postdocs, John Sulston and Bob Hurwitz, and they were working on very basic things in the worm, just looking at the number of cells the worm had, how it developed, how many cells ended up in the various compartments and where they came from. And they were doing very simple experiments to track the birth of cells during development and to establish the lineages, which cells ended up in the pharynx or the gonad or, the ovary and so on, and or the primitive brain that the worm has. And they noticed something very odd. They noticed that during development of the worm, there was about 10% of the cells died, and they always died at the same times and the same places. And later on then, when Bob Hurwitz left the Brenner lab, he decided to conduct genetic screens to try and dissect how programmed cell death in the worm really worked. So here's an example. This is a worm in the very early stages of development. Here you're seeing the production of cells, the early divisions in the worm. It's in the egg case here now. The early divisions are reducing the size of the initial zygote. And then you'll see these little cells that are kind of disc-like, very smooth, that appear, and they go in and out of the field transiently. And now the worm is moving around in the case, and you can't see a lot. But I'll show you, something that illustrates it a little better in a moment. So that's development in the worm. And they noticed that of the 1,090 cells born during worm development, 131 of those cells always died at the same times and same places. And so they published a number of papers earlier, just establishing lineage maps showing when cells were born and then what tissues they ended up in, and also showing when they died. And this study clearly was a very nice example of, programmed cell death where cells died in a predictable manner during development. And then Hurwitz did genetic screens to identify genes that underpin these programmed cell deaths, and identified, in fact, about 11 different mutants that he described, SED mutants, cell death defective mutants, that had either too little cell death, so basically mutants where all 131 cells that normally died during development didn't die, or embryos that had excessive cell death, so extra cells than the 131 cells died, or also mutants where the cells died as normal, but they weren't removed as normal. And in fact, this illustrates an engulfment mutant where basically the programmed cell deaths persist in the worm. They're not engulfed and removed by phagocytosis as they normally would. So as a result of a lot of these genetic studies and crosses, a basic framework for the regulation of programmed cell death was established in the worm. And that framework didn't really give any idea as to how these genes worked. All it suggested was there were two genes that were essential for initiating cell death, and one of those genes was SED3 and the other was SED4. So they promoted cell death in the worm. So if you had a SED4 knockout worm, you got none of the cell deaths in development. And then a gene called SED9, which seemed to block cell death during development. So a loss of function mutant in SED9 gave you catastrophic cell death. You got greatly accelerated cell death. Or a gain of function mutant in SED9 blocked cell deaths during development. And then this gene here that they discovered in another screen, an egg-laying defective screen called EGL1, seemed to also promote cell death by inhibiting SED9. So it's going to see this basic framework has been conserved in flies, in mammals, and of course it's been elaborated on and it gets much more complicated, and there's also some very important exceptions to how it works in the other organisms. But broadly speaking, this is the worm is kind of a tiny human in many respects, with some important exceptions. Now, again, I must emphasize, it wasn't like Horvitz worked out all these genes in the worm, and then, we then went and found the mammalian homologs and found that they, worked in programs. That wasn't like that. In fact, as we're going to see, SED9 is homologous to BCL2 family proteins. And before the SED9 was even sequenced, it had already been discovered that human BCL2 blocked programmed cell death in mammalian cells. And that was already known. And in fact, so that predated the discovery of SED9 in the worm. So SED3 is as we're going to see, it's a caspase, it's a protease. And in fact, the first human caspase called interleukin-1 beta converting enzyme had been discovered before the sequence of SED3 had been actually identified. So in fact, the Horvitz lab discovered SED3 was a protease as a result of searching the DNA databases at that time and discovering that SED3 was homologous to caspase-1. So, but both systems really cross-fertilized each other, where discoveries in the worm informed, mammalian system and vice versa. And there was this kind of back and forth through the different systems that really helped to kind of elucidate, what was going on. Now, here's how it all works. So it turns out that SED4 really is a platform molecule, a molecule that exists to form a platform for activation of SED3. And SED3 is a protease, it's a caspase. And so normally SED3 is inactive when it's in the cell. And in order to get activated, it needs to be oligomerized by SED4. And SED4 seems to be a constitutive dimer, and SED9 binds to it and keeps it from oligomerizing. And so the key to getting programmed cell death in the worm is to displace displace SED9 from SED4. And EGL1 achieves that by binding to SED9 and displacing it from SED4. And then when SED4 is free of SED9, it can oligomerize into an octamer that forms a platform for binding and activating SED3. And when SED3 is aggregated in close proximity like this, SED3 now gets activated and it goes on to promote programmed cell death. So that's essentially how the worm works. You want activation of the SED3 protease, and in order to do that, you need to cluster the SED3 protease on an activation platform, and that's what SED4 provides. But normally SED9 restrains the oligomerization of SED4, and then EGL1 binds to SED9 and displaces from SED4. Now, I will emphasize that although we know that activation of SED3, the worm protease, promotes cell death, there's actually, the worm is really bad for doing kind of molecular biology type studies. So there's actually very few substrates for SED3 known. So in the detail, it's not really understood how SED3 kills worm cells. Now, Bob Horvitz and John Sulston and Sidney Brenner received a Nobel Prize in 2002 for really, it's a worm prize, really. The prize was technically given for programmed cell death, but I think many people feel that the prize was more for the pioneering use of the worm, which of course helped to elucidate mechanism programmed cell death. But as I've said, many very key developments were made, or discoveries were made in mammalian systems, and in fact they informed the understanding of how the worm system worked. So let's look at the fly for a moment. I'm not going to dwell on this. The flies are similar in terms of regulation of programmed cell death, but actually very different in particularly one notable way. So we again have SED3 homologs in the fly. Now we have 6 of them. So SED3, the worm caspase or the worm protease, there's basically one protease in action in the worm. But in the fly now we have 6 proteases. Not all of them are involved in programmed cell death, but several of them are. We have a SED4-like molecule, which is called DARC, or also ARC, and that's has a homolog in humans as well. And that's the SED4-like molecule activates a a molecule, a SED3-like molecule called DRUNK. So the fly SED4, which is called DARK, binds to this molecule here, DRUNK, to activate it, and then you get activation of some of the downstream caspases. But surprisingly, although there are two SED9-like molecules in the fly, they're not important for the regulation of programmed cell death. They play no role, at least that has been ascertained to date, and that's been looked at very closely indeed. Instead, we see a new category of programmed cell death regulators, which seem to dictate the onset of programmed cell death in the fly, and they're called inhibitor of apoptosis proteins. And we'll see also they play an important role in mammalian programmed cell death. And then a new class of triggers of programmed cell death. Remember, in the worm, we saw EGL1 or EGL1 neutralize SED9, whereas now we see that these molecules called Reaper Hidden Grim and Sickle, and they're called IBM molecules, IAP binding motif containing molecules, and they're the key to getting the initiation of programmed cell death in the fly. Those molecules bind to the IAPs and they trigger degradation of the IAPs, or they displace the IAPs, and the IAPs are essentially the key to keeping the programmed cell death machinery off in the fly, because it seems to be in that DARK and DRUNK, they're inhibited by one of the IAPs, DIAP1. And so the fly cell death machinery, although it's broadly similar, that is kind of an important difference. SED9 homologs don't keep the fly cell death machinery off like they do in the worm. And instead you have IAPs now that are instrumental or key to regulation of cell death in the fly. So DIAP1 binds to DRUNK, prevents DRUNK from being activated by DARK. But then one or more of the IAP binding proteins can bind to DIAP1 and displace it and trigger its degradation. And now DRUNK can bind to DARK, and similar to the apoptosome that we heard about earlier in C. elegans in the nematode. Again, you form this apoptosome-like structure comprised of multiple caspase molecules that are now activated by binding to this activation platform, comprised of a SED4-like molecule, which is called DARK in the fly. And this is a cryo-electron microscopy structure. So the apoptosomes, both in the nematode, in the fly, as well as in mammals, have a very similar symmetry and a very similar structure. You have this disc-like pattern, this platform that aggregates multiple caspase molecules. And so that is definitely a highly conserved structure through these different organisms. So moving on to mammals, broadly speaking, mammals are basically a complicated worm. Again, we have caspases being activated by a SED4-like molecule, which in mammals is called APAF1. And that's restrained by a SED9-like molecule, which now, is the BCL2 family. So BCL2, BCLX, BCLW, MCL1, A1. We have 5 or 6 members of this family that are capable of blocking cell death. And then we have a family of EGL1-like molecules that in mammals are called BH3-only proteins. Now, there's an important other subset of the family we'll get to in a moment, that again, although this is broadly similar to the worm system and the fly system, there are very distinct differences. We're going to see that mitochondria play a very important role in the onset of programmed cell death in mammalian systems. And that is not the case in the worm and is not the case in the fly. So again, it's similar, but with distinct differences. And so looking at it in this way, we have the same framework. We have a whole host now of SED3 homologs. We have caspases 1 to 14. There's 12 caspases in humans, and I think 11 caspases in mice. We have one SED4 homolog APAF1 that's involved in activation of one of those caspases, caspase 9, to form an apoptosome that we saw in the fly and the worm. We have several IAPs, and we have maybe a couple of IAP binding proteins like we saw on the fly. And now we have a whole host of SED9-like molecules. And an important subset now that we see, which are called the BAX-BAC proteins. And they play a very important role in the onset of programmed cell death in mammalian systems by permeabilizing mitochondria, as we're going to see in a moment. So, broadly similar, much more complicated set of genes, but the same basic genetic framework as we've seen in the worm and in the fly. And mammals have an apoptosome very similar to the nematode and Drosophila apoptosome, where you have a SED4-like molecule APF1. It binds to a SED3-like molecule caspase-9. It forms this platform. And then once caspase-9 is aggregated on the platform, it now gets activated and it can go on to initiate a cascade of other caspase activation events, resulting in apoptosis. So broadly similar structurally to the fly and worm apoptosome. It has 7-fold symmetry. And again, you get the aggregation of caspase-9 here on the top to form these clusters. So this is bringing us up to date now. And in mammals, the programmed cell death machinery is obviously a bit more complicated than we've seen in the fly and in the nematode systems. And of course, there's lots and lots of things that can trigger programmed cell death in mammals. And these can be kind of organized into 3 major pathways, which I'm going to just briefly overview the details of. I'm near the end now, but I just want to check here, Gustavo, will we take a break for a moment or two, or will I just go through to the end and, finish up, or will we take a break for a couple of minutes?
We have a couple of questions, but
okay.
we should finish your presentation and end the discussion in 25 minutes.
Yeah.
So I think now it's better if you just finish, you wrap up, and then we start discussion, and then we make a break, few minutes, 15 minutes or so, before Christina starts with her presentation.
Well, let me wrap up then, because obviously people are watching this, and I feel, speaking online is obviously, it's difficult to gauge the reaction, see how tired people are and so on, whether they're at information overload. But so what I'll do is I'll just kind of quickly summarize the last few slides that I have in 5 minutes, and then I'll take some questions if anybody's still awake at this point. So what I was going to say here is that in mammals, we see that the programmed cell death machinery has been kind of elaborated upon, and we can collapse down essentially, all things that trigger apoptosis into 3 major categories. And they're often called, so if you read the reviews, you'll often see that there are 2 major pathways to apoptosis, the extrinsic pathway and the intrinsic pathway. But that overlooks a very important 3rd pathway, which is the Granzyme or CTLNK pathway to apoptosis. So the extrinsic pathway is a pathway that's initiated by death receptor engagement, as exemplified by the TNF receptor or FAS receptor. And here what we have is cell surface receptors that bind their cognate ligands, and then through adapter proteins, they bind to caspases. Caspases get activated, and then they either directly amplify and process downstream caspases to result in activation of a whole suite of caspases, or they can be routed through the mitochondria via cleavage of a BH3-only protein called BIT. And then pretty much all forms of stress and damage, radiation, cytotoxic drugs, toxins, and so on that kill cells can be put into the intrinsic pathway. So we have death ligands such as TNF and FAS and TREL. These are natural proteinaceous molecules that the immune system produces. They bind to cell surface receptors. But then any kind of injury or stress or damage to the cell, what that seems to do is activates 1 or more BH3-only proteins. And Christina will talk about this, I think, a lot more in her talk. And these are members of the BCL2 family, and they're EGL1-like proteins. But they do something very different to EGL1. So in some ways, they're similar. So they can bind to pro-survival BCL2 family proteins. So they can bind to the SED9-like molecules. But remember in the worm, I told you that SED9 binds to SED4. So the worm apoptosome is made of SED4 and SED3. And SED9, the worm BCL2, can bind directly to the SED4-like molecule. But that's not the case in mammals. And it's not the case in flies either, as I told you. The SED9 homologs in flies don't have any role. But in mammals, the SED9 homologs block the opening of this channel on mitochondria that's comprised of Bax and BAC. And really, that's very surprising. So a key checkpoint in cell death in mammals is the opening of a pore or channel comprised of the Bax or BAC molecules that, when they're bound by BH3 only molecules, that triggers their oligomerization. So if you get stress or injury that activates one or more BH3 onlys, they bind to Bax and BAC and activate them. And then cytochrome C is released from mitochondria, and cytochrome C acts as a very important cofactor for the assembly of the mammalian apoptosome. So, and then the pro-survival BCL2 families, the SED9 homologs, they block the opening of the Bax or BAC channel. So that's a very important distinction between how programmed cell death works in mammals versus the nematode or the fly. And then finally, we have the very important CTL-NK pathway, where these cells have cytotoxic granules in them that they can degranulate onto the target cell. They contain perforin, a pore-forming protein that permits the escape of the granule constituents into the target cells. And one of the most important granule constituents is an enzyme, protease called granzyme B. And granzyme B can also process and activate caspases, and can also process this BH3 only protein BID, which can open the Bax-BAC channel. So they're the three major routes to apoptosis. Pretty much all stimuli that have been identified in mammals that trigger apoptosis do so by activating either the extrinsic death receptor pathway, the intrinsic or BH3 only pathway, and then the granzyme B or CTL-NK pathway. Now, I'm not going to go into the details of what caspases do and, exactly, which caspase does what. There's lots of reviews out there on this, but it suffices to say that there's a suite of caspases. They all have different prodomains, which enables them to bind to different activation platforms. The reason they're called caspases is because they all have active cysteine at their active site. And they're all specific for aspartic acid residues. So they cleave their substrates after aspartic acid residues. And that's what achieves the apostatic phenotype. Now, they can be divided into pro-inflammatory, a subset that has a role in inflammation, and a subset that has a role in apoptosis. But really, I think of these are all inflammatory proteases. They either promote inflammation, so the caspase 1 or ICE subfamily promotes inflammation in the context of programmed necrosis. And then the anti-inflammatory category promote apoptosis. Because as I told you, I think the main raison d'etre to have apoptosis is in order to avoid inflammation. So that when you kill cells in a programmed manner, you avoid inflammation by facilitating the uptake of these cells early on by phagocytes so they don't release DAMPs and promote inflammation. And we wrote a review on this a number of years ago on Molecular Cell, if you want to take a look at that. But essentially, in that review, I proposed that caspases are involved in programs so that not to kill, but really to extinguish the inflammatory properties of cells so that they don't provoke inflammation. So we used to think of caspases as either being involved in cell death or inflammation, but I think of them as all being involved in inflammation, and they're either pro-inflammatory or anti-inflammatory. Now, caspases are typically activated in apoptosis by this cascade-like manner through cleavage at their active site loops. So they have these disorderable loops near the active sites that blocks the active site of the caspase. And then when you get proteolysis of these loops, now you open the active site so that the caspase can cleave its substrates. There's a lot of caspase substrates that have been identified in mammals. I'm not going to go into any detail on this, but really the point I want to make is that when caspases get activated during apoptosis, they cleave hundreds of proteins in the cell. And you're probably achieving a number of different things by doing this. You're inactivating certain inflammatory molecules, you're triggering the externalization of eat me signals and find me signals that allow phagocytes to recognize and engulf apoptotic cells. You're hydrolyzing DNA, I think, to facilitate its kind of easy removal. DNA is a very, viscous substance that, if released from a cell, is very difficult to deal with, and is, I think, a potent trigger of antibody generation. If that happens, autoantibody generation. DNA is also toxic. Histones have been frequently reported to have toxic properties. So you've got a kind of a coordinated demolition that's going on in the cell that's coordinated by caspases. And there's very few caspase substrates that are absolutely essential for the completion of apoptosis. And in fact, we don't even think that caspases in mammals kill. We think that they simply, they prepare the cell for recognition and disposal. And I'll tell you why I think that in a moment. But we know that, for example, the hydrolysis of DNA happens as a result of cleavage of a caspase substrate, ICAD, which normally inhibits an endonuclease called CAD. And then once CAD gets activated as a result of ICAD proteolysis, it then can go on and produce this endonucleosomal cleavage pattern that is typical of apoptosis. So there are hundreds of caspase substrates. We know that we've done a lot of proteomic studies over the years. And we used to think there were maybe a special subset of these substrates that if they weren't cleaved, the cell doesn't die. But after years of working on this, we came to the conclusion that that's probably not the case. And this is a review we published in Nature Reviews Molecular Cell Biology a number of years ago, summarizing some of the caspase substrates that make sense, that kind of trigger the membrane blebbing and the nuclear condensation and so on that happens during apoptosis. But I now think of caspases when they're activated in apoptosis as a little bit like this. This is actually a photograph a friend of mine sent me after he was painting his front room and then left his two children alone with the paint can for about 10 minutes while he took a phone call in another room. And I think when caspases are unleashed upon the cell, they cause a lot of destruction. That much of which I think is got to do with kind of, cleavage of innocent bystander substrates that don't make a lot of sense in terms of, what facet of apoptosis they achieve. But collectively, the end result is, the facilitation of this removal, this early removal of cells by apoptosis, so that they don't burst open, they're recognized and they're engulfed. And there's some evidence that there's an extinguishment of the inflammatory activities of apostatic cells. So some of the dams seem to be cleaved and inactivated by caspases during apoptosis. Now, I mentioned this fact earlier, and I'll just finish a couple more slides. I mentioned that mitochondria play a key role in mammalian apoptosis. Christina will tell you more about that. It's very surprising. It's really true. In mammals, cytochrome C release really is the point of no return in apoptosis. So once you get opening of the Bax-Back channel, the cell is really doomed to die. And why I mentioned that point is that's triggered by BH3 only proteins and Bax-Back proteins. So it's nothing got to do with caspases. Once you release all your cytochrome C, even if you block caspase activity downstream, you don't rescue cells. Cells don't survive. So you don't need caspases to kill cells. That's often a misunderstood point about mammalian apoptosis. Caspases don't kill in most pathways, but they convert the phenotype. They ensure the phenotype is an apostatic phenotype. And we know that because if we block caspase activation downstream of cytochrome C release, we still get cell death, but the cell death happens in a caspase-independent manner. It's necrosis. You convert, if you have cell death without apoptosis, so even if it's driven by BH3 proteins, if you open the Bax-Back channel and block caspases, then you convert the cell death to necrosis. So we think that caspases are required to ensure that the cell death happens in this manner characteristic of apoptosis, with all those features that kind of minimize collateral damage to neighbors and release of dams. So that's why we think we need caspases. We need caspases not to kill, but to control inflammation. And I will, I think, finish up on this. So that's really the take-home message. When you get cell death, you get caspase activation to coordinate all of the events that ensure that dams are not released and that apostatic cells are eliminated in an immunologically silent or anti-inflammatory manner. whereas necrosis is actively driving inflammation through release of DAMs, which, as I mentioned now, are molecules that are capable of actively engaging the immune system. And I think I will leave it at there. And some of the information I covered in this is covered in Essential Immunology. If you're interested in, I would suggest you take a look. Okay, I'll leave it there. Thanks, Gustavo.
Thank you, Seamus, very much. It was, again, a brilliant lecture. I know everyone in the room is very excited. We have some questions. I think Karina will start asking you. I think if you will stop sharing the screen, then maybe
okay.
We get a big image of yours through the screen. Karina?
Yes. Thank you, Shay, for this amazing lecture, as usual. So Diego will ask about the IL-1 family role as DAMs. So he asked, HMGB1 and ATP are classic DAMs because they act as finding and activation signals. Could you tell details about IL-1 role as canonical DAMs as well? So this is the first question, and I take the opportunity to have my personal questions about the role of the gazamine D and LKL role as to release these IL-1 related cytokines. Since you mentioned that only dead cells are able to release those cytokines.
Yeah. It's quite a big question, so I'll deal with the second part, MLKL and gasdermin. So I think these are pore-forming proteins that serve to, very actively and very rapidly release IL-1 family members when there is the kind of detection of a pathogen in the cytoplasm of a cell, or if a pathogen tries to interfere with the programmed cell death machinery. You can get a shunting of the programmed cell death machinery to the programmed necrosis machinery. And so I think we see now an emergence of a paradigm where there are certain scenarios where you can get programmed necrosis that is designed as pore-forming proteins, such as MLKL or gasdermin D, that are designed to rapidly burst open the cell and dump a huge payload of IL-1 family members very rapidly. And so I think everybody agrees IL-1 beta is a really important, I would consider it DAMP. And I think pyroptosis is a way of very rapidly getting IL-1 beta out of the cell. There's been a debate over the years how IL-1 beta gets out. We've contributed to that debate. We believe that the major mechanism of IL-1 beta release is through necrosis. Other people have published reports suggesting that, it may be able to get out of viable cells. I have to say I have my doubts about, whether that is ever a significant mechanism of IL-1 beta release. So I think gasdermin D and MLKL are great examples of when you want a very rapid immune response in the context of the detection of a pathogen, then you deliberately trigger necrosis via those molecules. And so, yeah, that's a great way of dumping IL-1 family members very rapidly. And you also dump caspase 1 out of the cell. And I think a lot of the processing of IL-1 beta actually occurs, post-mortem after the cell has released its IL-1 beta and IL-18 and so on. But going to the issue of DAMs and IL-1 family members, I've given some of the evidence. I mean, rather than go through it here again, I wrote a review a couple of years ago, actually in the FEBS journal, talking about, why I believe the IL-1 family members are canonical DAMs. I think they have all the characteristics that you'd expect of a danger signal. They are cytokines. They have a safety catch. You know, they need proteolytic processing for activation. There's really no evidence that they're released from viable cells, just through necrotic cells. And also, we know that, for example, there's very few diseases where HMGB1 crops up as being a problem, or ATP crops up as being a problem. I'm very dubious about whether HMGB1 is really a DAMP, to be quite honest. I don't think ATP is at all. ATP can activate immune cells, but ATP is a very good driver of necrosis. So that's how I think ATP activates immune cells. It causes necrosis that releases DAMPs. And, you know, we all know that ATP is a good kind of trigger of inflammasome activation. So, you know, I think IL-1 family members really are the key DAMs. We know that they have receptor antagonists. We know their receptors. We don't really know the receptors for a lot of the other proposed DAMs, or a lot of the other proposed DAMs like HMGB1. There's been plenty of studies suggesting that HMGB1 has no activity if there's no LPS in the system, you know, or, you know, some studies suggested it binds nucleic acid and it has no activity, there's no nucleic acid around. And there's been also debate about whether it's active in its oxidized form or unoxidized form. Whereas, you know, nobody debates the activity of IL-1 beta or IL-1 alpha or IL-33 or IL-36. I mean, these are cytokines. And whereas I think a lot of the other proposed DAMs are, should I say, a bit wafty, you know? They lose their activity depending on whose lab they're in. And, you know, I would much more favor IL-1 family as being really DAMs than some of these other molecules. GP96 has been suggested to be a DAMP, but again, you know, lots of people don't find that activity. And, you know, so I think there's non-classical or non-canonical DAMs, which I think are a bit controversial. And then classical DAMs are the IL-1 family members, which I think we all agree on. These are cytokines, and they're largely released mainly by necrosis.
So Seamus, the next question is about the separation between apoptotic and necrotic. That apoptotic is always silent, necrotic is always inflammatory. So there's three topics that go together here, if you can touch quickly on these three. So the first is, what if apoptotic cells are infected? The second, can pathogens take advantage of non-inflammatory cell death? And the third, can you think of a situation that necrotic cell that is anti-inflammatory?
Of a necrotic cell that isn't inflammatory?
That isn't.
All right. So, of course, the idea that apostatic cells are non-inflammatory, I think, is an approximation. And, of course, there are some very important caveats to that. So I think apostatic cells are generally less inflammatory than necrotic cells, is, I think, a more correct interpretation. Because, for example, we ourselves have published, and we know that molecules such as TNF can induce apoptosis. And TNF is a very important pro-inflammatory molecule. But it's less inflammatory than the alternative. So what I mean by that is you in fact can switch certain modes of cell death. We know you can switch TNF-induced apoptosis. You can switch that to TNF-induced necrosis. And I think typically when you switch TNF-induced cell death to necrosis, it's more inflammatory than TNF-induced apoptosis. So, you know, that there are scenarios, of course, where like FAST can induce cytokine production. That is inflammatory, but it's less inflammatory than FAST-induced necrosis if you switch to necrosis. So I think broadly speaking, yes, it's true to say that apoptotic cells are not totally non-inflammatory, but I think they're less inflammatory because you don't get DAMP release than the alternative outcome of necrosis. Again, it's kind of it's a point for maybe the people who know quite a bit about the cell death pathway, but we know now we can toggle outcomes between necrosis and apoptosis depending on whether we block Caspases or not. And I think the general pattern you see is when you get necrosis, whether it's programmed necrosis or just straightforward necrosis, that is more inflammatory than regular apoptosis. So in terms of the exploitation of apoptogens, Yeah, I think that is a very fertile ground for pathogens to exploit. I think generally speaking, pathogens try to interfere with cell death outcomes because it's in the interest of a pathogen to keep the cell alive for as long as possible. And there seems to be this molecular arms race, you know, in higher organisms where every kind of weapon that the pathogen develops to interfere with the programmed cell death machinery, there seems to be a counter strategy employed, you know, in order now to shunt the outcome to an even more aggressive form of cell death or a more inflammatory form of cell death. I don't know which is more beneficial. You could argue, you know, it's certainly not in a pathogen's interest to block apoptosis that results then in programmed necrosis, because the cell dies much more rapidly and induces a much more aggressive immune response. So, you know, I mean, people say this, oh, you know, in order to counter the fact or the effects of pathogens that encode caspase inhibitors, you know, the programmed necrosis machinery developed. Well, pathogens aren't stupid, you know. So, you know, they're selected in a Darwinian way. So if programmed necrosis was bad for a pathogen, then pathogens wouldn't trigger it. So, you know, it would just be selected against. So you could argue as well that in fact pathogens may deliberately interfere with the cell death machinery in a way that triggers programmed necrosis in order to wipe out immune cells very rapidly. That you know may in fact because we know that, for example, if, you know, TNF signaling can be shunted to programmed necrosis if you block caspases. But of course, you know, if a pathogen blocks caspases, but it's not in the context of a TNF signal, then the cell is going to live. So a good way to maybe just eliminate TNF responsive cells or cells that make TNF is to trigger programmed necrosis. So I think there's this arms race going on between pathogens and how they tinker with our cell death machinery for their own benefit. And I think it's context dependent to say whether it's better to have apoptosis or better to have necrosis for a pathogen. I really think it depends on what cells you're talking about. If I was a pathogen that affect macrophages and also some other cell types, I'd want to kill all the macrophages if I was a pathogen, and maybe all the T cells, because, you know, very quickly that's going to lead to a severe disablement of the immune system, which is in the pathogen's interest. So I think you can work this both ways, but it is an approximation or a crude approximation to say that apoptosis is always non-inflammatory. I want to emphasize it is generally less inflammatory than the alternative outcome if you had necrosis.
Karina, I think you have another question or from the audience. Some of the questions from the audience is very much related to apoptosis. So I think we should wait until Chris finish her talk, and then we may come back to those questions. One of those, Christina has even answered in the chat, but we can, you know, talk about that later in the discussion time of Christina's talk. I'm not sure if Karina has something else, or Ricardo, or because it's already 10 o'clock, we should go for a break. So no more questions now.
No more questions. Only congratulations for your amazing and very didactic lecture.
Thank you.
Right.
I wish it felt amazing sitting in a chair here in Dublin. I'd much rather be sitting or in a lecture theater in Sao Paulo, but hopefully soon.
Of course.
We miss you here as well, Shay. So hopefully, yeah, soon enough, we will have another version of this course presentially here in the University of São Paulo or the Einstein Institute or Unifesp. Yeah, so thank you so much, Shay. A lot of comment on your excellent didactic. So it was very clear to our students. We also, it's worth mentioning that the people from around the world, we had almost 100 people in the room. I'm gonna call now for a break, 15 minutes break. So now for me it's 10:05. So we come about 10:20. So you people that are watching at the YouTube, we're gonna turn the screen off, but don't go too far away. In 15 minutes, we are back with the new streaming of lecture number 2 by Cristina Munoz Pinedo, Apoptosis and Cancer. All right. Thank you very much for listening. And now I am turning